Okay, hey guys. So hope you're enjoying the snow that's out there. This is a video recording of the lecture that I was planning on giving today in class, wrapping up what we needed to talk about regarding communication of neurons, action potentials, and things like that, which we pretty much wrapped up discussing in class last week on Thursday. Just a couple more things to say about that, and we will get into talking about some of the more stuff in chapter 3 here. So let's just sort of get started here and recap a little bit on some of the things that we said last class, just to refresh your memory. So we were talking about neurons communicating. Let me get a pen going here. Neurons and how they communicate, and we said that neurons communicate through an electrical chemical process. The electrical part of that process is the action potential that travels down the length of a neuron's axon. So the axon, remember, is the output from a cell. The dendrites are the input to the cell. This particular neuron that we're looking at here doesn't necessarily have dendrites because this is a sensory neuron. In particular, it's a touch receptor, so it responds to the sensation, stimulus being some sort of sensation of touch, like pressure against your skin. So if you experience some sort of pressure against your skin and this receptor is below that part of your skin, that will, if the stimulation is large enough or intense enough, cause an action potential. It will stimulate the cell, which will mean that it will cause an action potential to travel down the length of this axon, right, away from the cell body to the synapse here. So we've said that neurons form synapses with other cells. They don't actually touch or connect with one another, at least not usually, right? So if the axon is the output from a cell, carrying messages away from the cell, the dendrites, which are these tree branch looking things here, are the input to another cell. So here's showing just a simple what's called neural circuit, which is just sort of a large number of neurons working with one another, but showing how this sensory neuron synapses onto this what's called a multipolar neuron. It's got lots of dendrites and just one axon coming off of it. And so here's just a blown up image there, looking at the axon of this cell. This would be the presynaptic neuron, since it's the cell before the synapse being the space, the space between these two neurons. So this is the axon of the presynaptic neuron, and this is the membrane of the well, it looks like we're actually not looking at a synapse onto a dendrite. This is looking at a synapse directly onto the cell body of this, what is called the postsynaptic neuron. And so we said before, when talking about neurons and how they communicate through an electrical chemical process, the action potential, which travels down the axon, is the electrical part. And when the action potential reaches the end of the axon, this initiates the second step in this communication process, which is the chemical part, involving the release of neurotransmitters from the axon of the presynaptic cell into the synapse, which get picked up, or at least potentially get picked up, by the postsynaptic membrane. So here you can see these little red things here are all neurotransmitters. And so neurotransmitters are contained in these what are called synaptic vesicles, little pockets that contain neurotransmitters that migrate over to the inside of the wall of the axon here, and will open up and spill their contents out into the synapse. And so all these little molecules of neurotransmitters get released out into the synapse, where they will potentially bind with receptor sites that are located in the membrane of the postsynaptic cell, which may or may not initiate an action potential in the postsynaptic cell. And we said last time that whether or not you see, just go back to that slide for a second, whether or not you see an action potential in this cell really depends on the message that's being conveyed by the neurotransmitters, and whether that message is an excitatory message or an inhibitory message. And so an excitatory message causes depolarization of the postsynaptic membrane, which means depolarization is that sort of first part of the action potential, right? It's the change in the voltage across a cell's membrane from what the cell starts out at, which is very negative, to being very positive. If remember, we drew the action potential here. This is time, and this is voltage that we're plotting as a function of time when a cell is at rest. The voltage across the cell's membrane is negative 70 millivolts. Oops, I don't want a highlighter. I wanted to just change the color of my ink here. So when a cell is at rest, it's got a negative electrical potential across its cell membrane. You can sort of think of it as like the electrical charge inside of the cell. When it gets stimulated by excitatory neurotransmitters and it initiates an action potential, what you see is depolarization, a really rapid increase in the electrical potential, or change in the electrical potential across the cell's membrane. It goes from being very negative to being, for a brief period of time, positive, and then back down toward the resting potential, and then actually overshoots it a little bit. If you remember, this is what we called the cell being hyperpolarized when it's in this state here, where it's actually somewhat more negative than when it started out. This is when the cell is hyperpolarized, and it takes a little while for the charge across inside the cell to get back up to its resting potential value. This whole thing is the action potential. So neurotransmitters that communicate an excitatory message make it more likely that the postsynaptic membrane will generate an action potential. So it increases the likelihood of an action potential. Okay. Whereas inhibitory neurotransmitters do exactly the opposite. These actually cause hyperpolarization. We'll look at that on the next slide here, making it more difficult for the postsynaptic membrane or the postsynaptic cell to generate an action potential, actually decreasing the likelihood of a cell generating an action potential. So this figure just sort of shows exactly that. Up here at the top here, we show the results, or the effect that excitatory neurotransmitters have on the charge inside of a neuron, right? So if you remember, we said last time that action potentials are sort of all or none events. They either happen or they don't happen. And whether they happen depends on whether the charge inside the cell gets past a particular threshold value. If the charge doesn't get past that value, you're not going to see an action potential. And the action potential is this whole thing, right? The whole up and down, depolarization, followed by repolarization, followed by hyperpolarization. Those three things are the sort of three major components of an action potential. So if the charge doesn't get past this threshold value, it won't elicit an action potential. But what does an excitatory neurotransmitter do? An excitatory neurotransmitter makes it more likely that the cell will generate an action potential, makes it more likely that the postsynaptic cell will generate an action potential. And the reason for that is because here is our resting potential value of our cell that's not been stimulated. When that cell picks up neurotransmitters with an excitatory message, those excitatory neurotransmitters cause a small depolarization. So increasing the charge, or bringing the charge inside the cell closer to that threshold value, but not quite to the threshold value. So that now what will happen, if this cell just gets a little bit more stimulated, right? Look at how close it is to the threshold value. It doesn't need much more to go before reaching this threshold value. So it's at this point, it's very easy to get this cell to generate an action potential, right? And if it just gets stimulated a little bit more, As this picture over here shows, you'll see that whole action potential of really rapid depolarization followed by repolarization, and it's not shown in this picture, but you should see hyperpolarization as well. Okay, so excitatory neurotransmitters do this. They cause a sort of a small depolarization of the postsynaptic cell, making it easier for this cell to generate an action potential. And why is it easier? Because the charge inside the cell for a brief period of time gets closer to that threshold value. It doesn't really have to increase much more to get to that threshold value. And so if you followed that, then you know exactly now what what an inhibitory neurotransmitter does. It does exactly the opposite. So here we start out at the resting potential value for this cell. If this cell receives or picks up an inhibitory neurotransmitter, that inhibitory neurotransmitter is actually going to hyperpolarize the cell, bringing the charge inside the cell away from that threshold value, right? So now look at how far this cell has to go before it reaches that threshold value, right? You need to actually apply a really pretty intense stimulus to this cell in order to now bring the charge from all the way down here to the threshold value to elicit an action potential. So this actually, an inhibitory neurotransmitter makes it much more difficult for a cell to generate an action potential. And those are the only two messages that neurotransmitters communicate, either an excitatory message or an inhibitory message.